Okay, good afternoon. So this is the Krebs cycle. Now on the top right of this diagram, you will see there the legend of this diagram. So for today, we are going to walk through one of the most important pathways in the cellular respiration. This is the Krebs cycle, or commonly called as well as citric acid cycle. So this is the stage where the cell fully breaks down what's left of the glucose to release high energy electron carriers that will later produce ATP. So let's go to the setting the stage. So remember um, that after glycolysis, uh, pyruvate enters the mitochondria and becomes acetyl coenzyme A. This one, acetyl coenzyme A, is our starting molecule. Now, the Krebs cycle happens in the mitochondrial matrix or in the inner fluid field space. So remember, glycolysis happens outside of the mitochondria. After releasing the pyruvate, as it enters into the mitochondrial matrix, it is converted into coenzyme A, and then the whole Krebs cycle happen in the mitochondrial matrix. This is the inner fluid field space. So think of this cycle like a circular conveyor belt. One molecule enters, a series of carefully controlled reactions happen, and at the end, the starting molecule is rebuilt so the cycle can continue. Okay, let's start. So acetyl coenzyme A, the first step is the acetyl coenzyme A, as you can see its structure, it has two carbons. And this is combines with oxaloacetate, which has four carbons, and together they form a six carbon molecule called citrate. So four from the what do you call this one? The four from the oxaloacetate, and then two additional two. So this is a key control point. So the cell speeds up or slows down this step depending on its energy needs. So next, the citrate is rearranged into isocitrate. Isocitrate through the enzyme aconitase. Nothing is gained or lost here. It's just a structural rearrangement to prepare it for the next reaction. Remember, when you hear the word isomerase, it means making an isomer. So it's not adding or removing something. It's the same content but different structure. So third is the isocitrate is then oxidized to alpha ketoglutarate. During this step, the molecule loses one carbon as carbon dioxide and produces NADH. So which carries high energy electrons to will you go na ba? Will you go na? Not yet. Okay, I'll catch up with you. electrons to the electron transport chain. So this step is catalyzed by isocitrate dehydrogenase, another major control point. Isocitrate dehydrogenase. So another carbon dioxide and another NADH, as you can see. So next is the alpha ketoglutarate. Alpha ketoglutarate um, is converted into succinyl coenzyme A. So again, the molecule loses one carbon as carbon dioxide here. It releases carbon dioxide, right? So that is actually a loss of one carbon. Okay, and of course, we also have another NADH form in this step. So this step uses the enzyme alpha ketoglutarate dehydrogenase, similar to the pyruvate dehydrogenase complex. Step 5 is the ATP. So for the ATP produced, succinyl coenzyme A is now converted into succinate. So during this transformation, the cell, all of that. Now, who else did not get? Thank you. If yeah, I remember. Oh yeah, okay. Thank you. So I was saying succinyl coenzyme A now converts into succinate. During this transformation, the cell produces ATP or sometimes GTP, depending on the tissue. So this is one of the rare cases of substrate level phosphorylation in the Krebs cycle. Next is the formation of FADH. Oh, it's not here. Mhm. Anyway, so succinate in here is then oxidized to fumarate by the enzyme succinate dehydrogenase. So this is unique because this enzyme is actually attached to the inner mitochondrial membrane. So during this step, we produce FADH, another electron carrier. This one. Oh, they just name it QH2, but that is FADH. So water is added. Fumarate becomes malate when water is added. And this step is catalyzed by fumarase. And it is a simple hydration step. When you say hydration, it's gaining of water. Next, um, so finally, malate is oxidized back to oxaloacetate by malate dehydrogenase and producing one more NADH. And with oxaloacetate regenerated, the cycle is ready to accept another acetyl coenzyme A. So for each acetyl coenzyme A that enters the cycle, the cell produces 3 NADH, 1 FADH2, 1 ATP or GTP, they are just cousin, and 2 molecules of carbon dioxide. So these NADH and FADH2 go to the electron transport chain where the real bulk of ATP is produced. So the Krebs cycle may look complicated, but it's really a repetitive system for harvesting electrons. If you understand the flow, combine, rearrange, take away carbon, collect electrons and regenerate, and then the entire cycle becomes logical and predictable. Okay, that's all for today, and thank you very much.